So people have been trying to take embryonic stem cells and grow them in a way in which you can isolate populations of hematopoietic stem cells, but not from the mouse, but actually do it in culture. And so you can do things with them. You could potentially use them in various applications. You could derive them from people with different diseases or conditions. You could use them for drug testing, for example. What people have mainly done is taken either the murine embryonic stem cells or human iPS cells and tried to culture them in different conditions in which you can try to reproduce what's going on and make them turn into populations of hematopoietic stem cells you can isolate. What we did is slightly different. It's known that if you take ES cells and you inject them into a mouse, they'll form what's called a teratoma, which is a tumor that has all kinds of different tissues in it. So Giovanni Amabile, my postdoc, thought, well, what if we take a human iPS cell and put it into a mouse, an immunocompromised mouse, and where it's known to form a teratoma? Can it form an environment that would then support the propagation, the development and propagation of hematopoietic stem cells? And so we cheated a little bit. We didn't go into culture because we're not smart enough to do that, but no one else is either. We let the mouse produce the environment that did that. And the paper's about using that technique to produce populations of human hematopoietic stem cells that you can then test by transplantation into an immunocompromised mouse. So there are three things that we did in the paper that were pretty interesting. One is that we were able to generate these cells and show by functional studies that they actually work as hematopoietic stem cells. The second thing that we did is we found that if you injected not only the iPS cells, but also stromal cells, a stromal line called OP9, which is actually a murine stromal line, that we could actually enhance the ability of the iPS cells to form hematopoietic stem cells. And the other interesting subplot in that story is you can take these stromal cells and you can put different genes in them to basically change what they make. And so we used different stromal lines that made different things and showed that some of them augmented hematopoietic stem cell production, some of them augmented the production of T cells, different lymphocytes. And so that's the second part of it, is that these stromal cells could enhance the ability to make hematopoietic stem cells, and you can actually alter their properties depending on what kind of a cell line you put in. And then the third thing we did is that we demonstrated that you could actually make hematopoietic cells, more mature cells that were actually functional. And so we made B cells that could make certain antibodies, human antibodies, because they're derived from the human iPS cells. T cells that had certain functions by making certain cytokines, and myeloid cells that could actually engulf bacteria. So we could show that you could make different lineages that contributed and were functional, but also it raised the possibility you might be able to make human antibodies this way. And so now a lot of people are trying to duplicate our results, and I've actually heard from at least one lab at the Harvard Stem Cell Institute where they've actually, at least, they've just initiated the studies, but it looks like they've been able to get the same kinds of results, which is very good.